Hello friends, I wish you remember the first page of genetics when we study. You said the field which genetics study on, one of them is the molecular structure of the genetic material, which now we're going to study on. Molecule base for genetic. Mendel, through his study for many traits in plant of peace, concluded that there are genetic factors. He didn't use the term of chromosome or DNA, but said there are genetic factors control the transferring the traits in organism from one generation to another. But what are these factors? What is the procedure to store the genetic information and its ability to solve the visual information which puzzled the scientists? Later, through their studies and researches, could solve some aspects of that puzzle by insistence of scientists to find a solution for one, the respiration system diseases, which was spread in society in 1928. Detection of DNA. Yes, there is some material which transports some trace between generation or something from one organism to another. But what is this material? It is the experiment of detection of DNA. Scientists depend on the three studies defined that the DNA is the genetic material which is transported from one organism to another, from one cell to another, or to the next generation through divisions. Created experiments on bacteria, so he explained that there is genetic factor which did the transformation. He could transfer the ability of killing between types of bacterial cells. As you remember from the conjugation in sexual reproduction of bacteria. The experiments of Avery and showed that the DNA, not the protein, are responsible for this transformation in bacteria. Then the experiments of Hershey and Chase, these two scientists in 1952 had the test to know if the DNA or the protein is the genetic material which the virus transfer it to the bacteriophage. You know the bacteriophage, the kind of virus which attack the bacteria equally, and most of information about the viruses in the past is gained from that virus. This experiment, to be sure about, it is the DNA which transported or caused changes, but not the protein. Explain the experiment in this 3 steps. Radiation was used to distinguish the DNA, while radiation to observe it easily under the microscope was used to distinguish DNA from the protein in virus. So radiated phosphorus used for DNA, phosphorus for DNA, and radiated sulfur for proteins. After that, the two scientists left the virus which contained the radiated phosphorus and one which contained the radiated sulfur individually to affect the bacteria. Which bacteria? E. coli bacteria. The layers of virus were removed from the cells by a certain processor. The virus were separated from the bacteria by using centrifuge, which turning the test tube very fast, and the molecular which high weight moved to the bottom, but low weight moved to the up of the test tube. The result was all viruses, the DNA a little bit protein entered to the bacteria. So when they observed the bacteria, they observed more radiated phosphorus than what? Than sulfur. It means more than protein, the DNA passed into the bacteria or increased in bacteria. On that base, it was concluded that the part of virus which affect the bacteria cell multiplying is the DNA, but not proteins. So phosphorus radiated, sulfur radiated. You know, virus has genetic material and protein code. So when the part of virus are removed, then they observed the bacteria and they saw it more than protein. There are genetic material or viral DNA. So they concluded that it is not protein, but it is DNA which affect the bacteria and multiply. Then discovering or detection of DNA, the studies of scientists go on about its structure. Until the year 1953, the biology scientists were proving the idea of DNA as the genetic material, but before that time they didn't know the structure of DNA. Then two scientists, Watson and Crick, you may remember from the first page, put an idea for DNA structure. It basically consists of two anti-parallel chains. Chains of what? Chains of nucleotides. One of them turns around the chains connect with the base which are related, like complementary. So two chains, one of them like base, but the second one is complementary chain. In the opposite chain by hydrogen bonds. How they connected? They connected one to another by hydrogen bonding. Also the sugar circles and phosphate group in each of chains by covalent bonds. Deoxyribonucleic acid DNA is regarded as a complex chemical compound. It is present in all living organisms and it's important. It's present mainly in the nucleus for eukaryotic cell within the chromosomes which forms from the chromatin reticulum, and chromatin material forms from the unit of nucleosome which consists of 4 molecules of histone, each one present in dual case. Nucleosome like a piece of protein but coiled genetic material DNA called a nucleosome. The DNA also represent in some cytoplasmic organisms in mitochondria and plastic which have their own DNA. Molecules of this acid, nucleic acids, is the biggest biological molecules, as polysaccharides are consist of number of units or small molecules which are monosaccharide. The proteins are consist of the number of units which are smaller molecules called amino acids. Like proteins made up of smaller parts, polymer protein, monomer amino acid. Polymer polysaccharides, their monomers are monosaccharides. And the nucleic acids, which are polymers, their monomers are nucleotides. Means how proteins made up of smaller units called amino acid. The nucleic acids consist of smaller units known as nucleotides. Each nucleotide, which is monomer of nucleic acid, consists of 3 simple molecules connected with each other directly. Its form out inside the follows, like 1 pentose sugar, pentose, which means 5, so this sugar have 5 carbon in its chemical structure, 5 carbon sugar. Deoxyribose, we see in this part, carbon 5. How we may remember the glucose from before, which have 6 carbon, 12 hydrogen and 6 oxygen, but pentose sugar has 5 carbon. Phosphate group, this consists of an atom of phosphate is connected by 4 atoms of oxygen. The alternate molecules for phosphate and sugar form the 2 sides of DNA chain, so the nucleotides which are on the way of each chain connected by covalent bonds combines between the sugar of one nucleotide and the phosphate group of the other nucleotide. As we said, there are 2 chains from that side. If there is sugar, it connects to the phosphate group to the other side by what? By hydrogen bonding, covalent bond. Its unit 2 atoms as a result of incorporation of each one of them by an electron. The sugar and the phosphate are identical in each nucleotide. It's important, the sugar and phosphate identical in each. All nucleotide in DNA, they all have pentose sugar, deoxyribose and phosphate group. Nitrogenous base is a ring compound which contains nitrogen in addition to carbon, hydrogen, oxygen, which except one of them. Adenine have not which does not contain oxygen. The distance between each base and another one is constant. So because in each nucleotide, the pentose sugar, the phosphate group is same, so the name of nucleotide depend on nitrogenous base, because only nitrogenous base diverse in nucleotides. But not too much. There are how many? 5 types of this nitrogenous bases, which divided into 2 groups, which is pyrimidines and purines. Pyrimidines like thymine, available in DNA only, cytosine available in both nucleic acids, because more than DNA we have another nucleic acid which is RNA. So thymine only for DNA, cytosine for DNA and RNA, but uracil only for RNA. The purines, adenine available in DNA and RNA, and guanine also found in DNA and RNA. These 5 kind of what? Nitrogenous bases. Because we said the phosphate group, the pentose sugar in each nucleotide of DNA is same, so the name of nucleotide depend on what kind of nitrogenous base. So before they ask question in ministry exam to explain structure of one nucleotide in DNA, we start from the pentose sugar, phosphate group, nitrogenous bases, and give this kind of nitrogenous bases. The linkage of the bases between 2 chains is not random, but it's bounded. So adenine in one of chain always connect with thymine. So adenine, which we symbolize by A, always it make pair with thymine in the other chain. So first base chain and complementary in base, if there is adenine, it's completed always by thymine. By how many? 2 hydrogen bonds. The cytosine in one of them connect with T, guanine, cytosine always completed with guanine, and with how many? 3 hydrogen bonds. So always A completed with T, T completed with A, C with G, and G completed with C. The scientists Chargaff integrate that the percentage of adenine equals the percentage of thymine, because always they complete with one another, and also percentage of guanine and number equal to number of cytosine in DNA for various group of organisms. And these pairs of bases are called complementary base pairs. So A and T, one pair, C and G, another pair. So the order of the bases in one chain of DNA molecule or RNA is complementing by the structure of the bases in the opposite chain. For example, the chain of DNA with the order of A, G, A, C, the opposite chain will be completed with A completed with T, G with C, A again with T, and C with G. So if in questions give out information about one of these chains, we can find the sequence of nucleotide or nitrogenous bases in the second or complementary chain. Complementary base pairs are important in DNA structure and function for 2 reasons. Because the bonds between the pairs helps to hold the 2 DNA chains, which are hydrogen bonds, and second, because of complementary nature for the DNA, have to explain how it is multiplying before the cell division. We know in interphase stage the DNA is duplicated. Example, if you know the succession of the base in one of the DNA chains, which is like the TC, TG, TG, GAC, while it makes groups of 3, I explain it after, how the complementary base will be in opposite chain. So for a solution, we write the given succession of nitrogenous bases, and then we complete it. We complete like T completed with A, G, C with G, T with A, G with C, T with A, G, C, C, T, and G. The above shows that the DNA is very important because it is the forming the material for the genes. So you may remember the mutation, any of these nitrogenous bases. Chain-like point mutation cause what? The sequence of nitrogenous base like frame shift mutation or displace mutation, and cause what? Changes in the phenotype of organism. The succession of the nitrogenous bases determines the traits of organisms. Replication of DNA. Yes, DNA is duplicated before division, cell division, mitosis or meiosis. In what? In interphase stage. The main property for the genetic material DNA is the ability to replication in the beginning of each cell division to make sure transferring it to the new cells. Helicase enzyme, like a scissor, we know this enzyme separates the DNA chain. How we said the two DNA chain are connected one another by hydrogen bonds. So these hydrogen bonds are broken and two chains are separated by what? Helicase enzyme. So this enzyme transfers on the length of the molecule to open the hydrogen bonds between the complementary bases. The product of separation, these two chains in an area looks like the shape of letter, white but upside down. So the DNA look like this, connected by hydrogen bonds. From that side, it starts to be separated one from another. And like in this way, as separated, these two chains move away one from another to rebuild new DNA and look like what? An upside down Y letter. The enzyme of DNA polymerase adds completing the nucleotides which are inside the nucleus to each one of the original chains, and of course the covalent bonds forms between the nearby new nucleotides. So the hydrogen bonds form between the complementary bases which are on the original new cells. How we said the helicase enzyme separate these two chains, move away one from another, but the polymerase enzyme builds new chains of DNA. So how the separation continue, building new DNA continue, but at the end two new DNA are built. Notice the direction of the arrows of this figure replication fork that building the DNA in opposite direction in each chain. This leads to make holes in the chain which is forming recently, but these holes get connected in between them by an enzyme called DNA ligase enzyme. The genetic material DNA in a nucleus or chromatin network is too long, about 46 chromosomes in human. It's about 2 meters long. So not only one helicase enzyme broke it down, but from the many points. And from each point, helicase enzyme followed polymerase enzyme to build new DNA. As the new DNA produced it like what? Like different pieces, 100 pieces produced by 100 enzymes. But how the pieces of DNA are connected, bonded to one another by DNA ligase enzyme. As building is completed, the DNA polymerase enzyme and the replication then separate from it and produce two separated molecules in each chain. One of them is original and the other one is new. So this kind of replication is called a semi-conservative replication. We look this picture, if now we can see two chain of genetic material which is connected one another, but from this side it start to be separated one from another by DNA helicase enzyme. So this point of separation we call it as what we can remember from this part, the replication fork. Look like a fork with a replication start from that point. And then the DNA ligase enzyme build new two strands. How we said, like A completed with T, G completed with C, by what? By DNA polymerase enzyme to build new pieces of DNA. But new build of DNA, at it go on forward, separated, followed by DNA polymerase enzyme, and new strands of DNA formed. When the building or replication, copying of DNA is ended, the DNA polymerase enzyme, to prevent the mutation, which is the changes happen in genetic material. This isn't the present, but we can see the mostly about billions of new nucleotides added, but about 100% or by low mistake it rebuilt because the DNA polymerase enzyme, which mostly repair the mistake. How it go on to build new DNA, then it move backward to control and check it if it is true. If there is some mistake, rebuild or repair or change the nucleotides. So in this part we have three enzymes. Helicase enzyme separate two chains of DNA. DNA polymerase enzyme produce new pieces of DNA, complete the separated chains by new one, and the DNA ligase enzyme connect the pieces of DNA one to another to make a long chain. But as a result, this strand old, but build a new one. So the new DNA at the end, half old, half new DNA, half new and half old DNA. So this kind of duplication or replication is called as semi-conservative replication, because half of DNA is preserved, not changed, it is old original, but half of it new built DNA, so it is semi-conservative replication.